Hello, everyone. I'm Linda Tintawi, CEO of the Lustgarten Foundation. Welcome to our annual year in review and outlook for 2026. Today, you will hear from the three leaders responsible for guiding and driving this progress. Lustgarten's Vice President of Research, Dr. Andrew Rakeman, our Chief Scientist, Dr. David Tuvison, and our Chief Medical Advisor, Dr. Elizabeth Jaffe. Progress in pancreatic cancer does not happen by chance. It is built deliberately through strategy, persistence, and an unwavering focus on patients. Last year, even as biomedical research faced serious challenges, the Lustgarten Foundation continued to move science forward, stand up for researchers, and mobilize a nationwide community united by one goal: turning pancreatic cancer into a curable disease. Here are some of the highlights which today's guests will discuss in greater detail. Our foundation remains laser-focused on transforming early detection and interception. In partnership with the Mark Foundation and the American Association for Cancer Research, leading scientists, clinicians, and regulators convened for a landmark, first of its kind, early detection workshop to identify how to move progress faster. Importantly, this has led to new funding for bold, cross-disciplinary research, earlier diagnosis saves lives, and collaboration accelerates progress. Throughout 2025, one theme stood out as momentum accelerated around new treatments: KRAS. This genetic driver is involved in more than 90% of pancreatic cancers and was once thought to be undruggable. New KRAS-targeted drugs are now in advanced clinical trials, including one supported by Lustgarten. This marks a real shift from skepticism to possibility and brings patients closer to more effective, targeted therapies. At the same time, Lustgarten-funded teams are tackling drug resistance, aiming for combination therapies that last longer and work better. These are encouraging signs for immunotherapy as well. Early studies of a KRAS-targeted vaccine showed strong responses and promising links to better outcomes. Building on this, Lustgarten is supporting next-generation vaccine trials and combination approaches designed to help the immune system recognize and attack pancreatic tumors. And finally, data is reshaping our decision-making. Results from our PASO-1 clinical trial showed how precision medicine can directly guide patient care. By matching tumor biology to treatment choices, this study demonstrated clearly and in real time that data-driven decisions improve outcomes and can change how clinical trials are designed going forward. We encourage you to ask your questions today. You can type them in the Q&A box below. And it's now my great pleasure to welcome Dr. Andy Rakeman, Lustgarten's Vice President of Research. Andy, we have so much good news to cover today, and I'm really looking forward to the discussion. So over to you, Andy.
All right. Thanks so much, Linda. And you're right, we do have a lot of good news to cover. And as you pointed out, this is not progress that's happened by accident or just because we willed it to be so, but this is the result of hard work, sustained work, and a commitment not only for the folks here on the screen and for the scientists working on it, but really from the whole community that's helped to drive this forward and keep focus on this. And who better to join me to talk about the great progress that we've made over this past year and really highlight where we're going in the future are the Lustgarten's Chief Scientist, Dr. David Tuvison, who is the Roy J. Zuckerberg Professor of Cancer Research and the Director of the Cancer Center at Cold Spring Harbor, in addition to his role as our Chief Scientist, and Dr. Elizabeth Jaffe, our Chief Medical Advisor, who is Deputy Director of the Sidney Kimmel Comprehensive Cancer Center at Johns Hopkins and co-director of the Skip Vraw Pancreatic Cancer Center, Associate Director of the Bloomberg Kimmel Institute for Cancer Immunotherapy. So Dave, Liz, so glad you could join us today. So glad you could be here to talk about the exciting progress that we've made, but also what's made that progress possible, where we're going in the future, and hopefully understanding how we got to here is going to help to guide the path forward from here and to really, as Linda pointed out, turn pancreatic cancer into a curable disease. So thank you for being here.
Well, thank you, Andy, for this nice introduction, and to Linda, and welcome everyone this morning or this afternoon or this evening, wherever you're dialing in from. Uh, as the basic scientist, as part of this organization, I've never been more excited about the basic biology of pancreas cancer, what we know about it, what we're learning by studying patient samples and model systems, and how the years of investment by hardworking scientists in academia and the private sector, supported in a large part by the Lustgarten Foundation, has led us to ideas that we could take to our colleagues who are really pushing the boundaries in the clinic. And that's my introduction to Liz, so she could now tell us how it's so exciting in the clinic.
Thank you, Dave, and thank you, Andy and Linda and all of the Lustgarten team. It's really been an honor to work with all of you on this very serious disease. So I just wanted to start by saying these are unprecedented times for patients with pancreatic cancer. I've been treating patients now since 1989, and it was terrible. It was terrible as a physician and even more terrible for patients. But we're finally at a time where we're seeing responses to new therapies. Until now, progress has been quite slow, as you all know, for decades, but the decades of investment in basic science, and Lustgarten has been a major investor in this science. This science is now driving new therapies. And this is critical. As you heard from Linda, KRAS targeted drugs are game changers. About 35% of responses are being seen even in patients who have failed chemotherapy with advanced disease, and up to 92% of patients are benefiting even with this advanced disease. So these are very important drugs. They're still in clinical testing. We expect them to work even better when we give them at earlier stages or before or with chemotherapy. KRAS targeted drugs still have some challenges we'll have to address with more basic science, but most patients should be able in the near future to try these drugs and hopefully benefit from them. We do need to figure out resistance mechanisms like all drugs. These drugs have resistance mechanisms, but again, basic science is already working to do this. My group works on KRAS targeted vaccines. We believe that getting the immune system involved early with these drugs can extend their durability. And there are other drugs coming as well. There's so many different findings in the tumor microenvironment because of all of the amazing technologies that we have, that now we're really able to start to develop specific drugs that are less toxic and more effective for more patients with this bad disease. I'll turn it back over
Great.
to Andy.
Yeah, no, thanks, Stephen. Leslie. A great way to get us started and really thinking about, and I, agree with the both of you that it feels like a different time. And I feel like we put in the decades of work where, back when this Lustgarten Foundation was started in the late 90s, we, really had hope, and that was about it. We turned that hope into promises to loved ones, promises to patients, promise for learning more. The promise turned into progress. And now we're on the cusp where progress is really starting to turn into impact and expectations that our patients and our loved ones are going to do better down the road. And I think I really hope that people take away from that about what it's taken to bring us along on that journey going forward. And you both already brought up these KRAS drugs that we hear a lot about in the news. We hear a lot of excitement about. And I think that's where we should start talking about progress over the year, because really, 2025 was a big breakout year for these drugs. We learned a lot of data in the clinic. We learned a lot about the potential, but maybe backing up a little bit on this idea of how did we get to here? And I'll start with you, Dave. Can you, just give the audience here a little bit of a sense of, why KRAS, first of all? Why do people keep hearing this funny name? Why was it so hard to target and address, and kind of what were the breakthroughs that set us on this lightning bolt of a journey we're on right now?
Sure. So in the late 1960s, the effort in the field was to try to understand the molecular basis of cancer. What causes cancer? We didn't know. We hadn't yet identified the the genetic causes behind cancer. We did know that there were certain viruses that promoted cancer, and much of the focus were on the viruses. When technologies were developed that allowed us to identify genes, human genes, one of the first genes identified was the RAS gene by groups of scientists around the country. And that happened in the very early 1980s, 1980, 1981. And so a gene which, when transferred from a cancer cell to a normal cell that included RAS, promoted cancer properties in the incubator and in experimental models. There were 3 RAS genes. One of them was called KRAS. KRAS turned out to be mutated in most pancreas cancers. We know it was the gene that was the accelerator because if you just transferred it into a normal cell, it caused cancer. The problem was KRAS in the gene, when it's mutated, it formed a protein which moves slowly. And the drugs that we have for most of medicine are drugs that slow down proteins. We don't actually have very many drugs that speed up proteins that make them move faster. And so we had a conceptual hurdle. Now, there were some really great scientists working on this, and they attempted to make it so RAS couldn't go to the membrane. And those experiments were very exciting, but in the clinic, they didn't work. And so we went back to the drawing board. And actually, the Lustgarten Foundation got involved in this project. So approximately 15 years ago, we got a proposal from a chemist at Harvard named Gregory Verdine, who had a new idea about how to target RAS. We decided to support Dr. Verdine. He was a professor at Harvard University on some of his exciting studies. Ultimately, this led Dr. Verdine to start a company. Ultimately, that meant he had to give up his professorship. And so he worked really hard on this problem. And I'm pleased to say that his early efforts led to another company acquiring his ideas and improving upon them, which is the best known RAS drug we have available today. At the same time, a chemist at UCSF named Keyvan Shokat developed a drug, a chemical drug that could inhibit one of the RAS proteins, which has a cysteine mutation at codon 12. Again, these are just fancy science terms, but a protein is like when you go to the store and you pick out grapes and you pick it up and you shake it and the grapes jiggle. Imagine each grape is an amino acid and that cluster of grapes is the protein. And that grape cluster does something. That's what your proteins in your cells are. And so the RAS gene, when it's mutated, one of the grapes, instead of being red, is now purple. And that makes the protein work differently. What Keyvan did is he developed a chemical that bound to the purple grape, and that inhibited the G12C version of the RAS protein that's important in lung cancer. That led to other companies making other drugs that look good. Pancreas cancer doesn't really have that mutation, though. And so Greg's early work led his idea to be purchased by another company. With Keyvan's insights, they were able to formulate a new drug, which is now the one that Dr. Rakeman talks about and Dr. Jaffe prescribes to patients. It's an amazing drug, but it's not the whole answer. And that's why the scientists involved in this need to work extra hard to find the extra things to make that first drug work well. There will be other drugs. But we were very excited about being involved in the ground level in that first RAS idea. It was so risky that a proper university didn't want their scientists to work on it. The government would not fund it. And the person had to essentially fail as a businessman. That's how many of the big breakthroughs in humanity are. There's a great idea, followed by failure, followed by resurrection. And that's where the RAS drug is today. So we're really excited. We're very excited because the scientists that we work with at the Lustgarten are working super hard to find ways to help when that first RAS drug doesn't work. And we are doing experiments, all of us collectively, and sharing our results with Dr. Jaffe and the clinical side of the organization so that the Lustgarten can fund deep science projects to push this forward. But I want to turn it over to Dr. Jaffe because she can describe how the Lustgarten has funded that first wave of science in patients using the RAS drugs and the very, I would say, creative way that we've done this, I think, for the betterment of all of you. Liz.
Oh, you're muted, Liz.
Yes, very sorry. So just to reiterate that the results have been really outstanding in this testing of the first drug that should be approved by the end of the year. And this is a Revolution Medicines drug. And this is in patients previously treated with pancreatic cancer who have one of several mutations in their KRAS. And the overall response rate is 35%, with a disease control rate of about 92%. That was the phase 2 data. They're now doing a randomized phase 3 study, and the data should be available pretty soon. What's also important is long-term follow-up. So the median progression-free survival, so stable before patients' tumors start to grow again, is only about 8.5 months, with an overall survival of 13.1 months. So this is important because, as Dave has said, we still need more science. And that's where Lustgarten is now putting a lot of their effort. And so they are funding studies, and they've partnered, as Andy has said, with a number of different groups, including Breakthrough Cancer, where we're helping to understand how these drugs are either working or causing resistance in patients' tumors. So you can imagine this very large phase 3 study that's ongoing that we should have an answer to soon. Well, a significant number of those patients are donating their pre-treatment and during treatment biopsies of their tumor to allow a group of scientists funded by Lustgarten and working with Breakthrough Cancer to really understand what are the changes in the tumor with this drug that either make a patient continue to have the tumor shrink or are preventing the patient's tumor from shrinking. And we're learning a lot about these mechanisms. And the hope is that we can now develop ways in which we prevent the tumor from escaping the drug's effects. Some of this is immune effects, and I'll talk a little bit more later on in the session about how we're trying to combine now immunotherapy with these KRAS inhibitors. And again, this is being funded by the Lustgarten Foundation, and we've been able to even learn how a vaccine targeting mutated KRAS can actually induce a patient's own immune system to fight their cancer. So again, bringing different areas of science, basic science together to really solve this very important problem. And that, of course, is pancreatic cancer.
Yeah, no, Dr. Jaffe, Dr. Susan, thanks. And I think we've heard a lot of the things that we've been talking about, this journey starting with a curiosity about biology in the world that turns into some focus that becomes driven by patients, that requires bringing in new technologies and new approaches, that has paths that don't work out the way that we want, and we backtrack and we keep working until we see some success. And I think we'll, over the next kind of advances throughout this year that we're going to talk about, I think we'll hear about the importance not only of developing new drugs, but clinical trial programs, understanding the biology moving forward. But before we get into that, maybe I'll ask you, Dr. Jaffe, just take a step back a little bit. You talked a lot about the impressive numbers that we hear about daraxonrasib, but the response rate and the control rate and the improvements in survival. But can you talk maybe a little bit more from what does a patient experience on these trials when they're on these drugs? What do those numbers really mean in terms of the life of the patient and what they experience during treatment compared to what the standard treatment had been for these heavily treated already patients?
Right. That's a really critical question, of course, because we could have lots of treatments, but if a patient doesn't feel well on those treatments, are they worth very much? So I could tell you that with chemotherapy, patients don't really feel great, right? Some patients do okay, but the majority really do not feel great. They might have a good week or two between treatments, but long-term effects include neurologic problems where their fingers become numb and painful. They can't even pick up glass, cups or glasses sometimes. So there's a lot of toxicities associated with chemotherapy for the benefit they're getting, which is really measured again in 8 months benefit overall. So that's a real issue. Whereas these KRAS drugs, now they do have toxicity, but they're more skin toxicity and a little bit of gastrointestinal toxicity. But patients overall feel so much better. We are able to manage those toxicities. Excuse me. And importantly, we're learning again, the science is telling us why these drugs are causing that toxicity. So we're learning a lot about the mechanism of how these drugs work, and we'll be able to really tweak these drugs so that we can hopefully even minimize the toxicities that patients may experience now with our future drugs. And that's really important because already there's a lot of science going on to help us figure this out. So I think we're in a good spot. I think things are accelerating very rapidly because again, Lustgarten has supported a lot of this research. When I started, 3 decades ago, and Dave could speak to this too, we did a study of how much pancreatic cancer is being funded by the NCI, the National Cancer Institute. It was minimal. There was so little. Lustgarten has been a critical component to making sure that not only are they funding research, but they're bringing so many people into pancreatic cancer research that was never in it before. So I think I just have to say that honestly, because for me, having seen this field, the only reason that we're moving so fast now is because of this investment and also the advocacy of the Lustgarten Foundation.
Thanks, Dr. Jeff. And so, Dr. Tuvas, I'll turn it to you a little bit. I think as stepping it back to the fundamental research side, what are the questions we're going to be curious about in the future? We heard from Dr. Jaffe about, these drugs don't work forever. Patients develop resistance. They develop some other toxicities around that. So the question is kind of is your lab and other labs like yours learning from what's happening in the clinic and taking that back to the mouse models to try to get to that next point, kind of in the Wayne Gretzky model, skate to where the puck is going, not to where it is, to think about, what are those questions that hopefully won't be 10 and 15 years down the road, but, 3 and 5 years down the road that we want to be addressing.
Yeah, no, so it's a very good metaphor you just used, particularly given the winter storm that we're experiencing in much of our country right now. And what we've been trying to do is build model systems that will predict the future, as you described. And so one that the Lustgarten Foundation really has established for the field was the development of tissue models, which we called organoid models, more than 10 years ago now.
Mm-hmm.
And these are small fragments of a patient's tumor that can be cultured. And you see the backdrop on the screen here, see the funny looking thing at the lower right with the bumps on it. That could be an organoid. And so that's an avatar of a patient's tumor. Those are the cancer cells. And if you had a dish of a patient's tumor, you could ask, what's the right medicine to wipe out this tumor? What's a medicine that, if you used it in combination with other therapies, would be the best? The reason we want to have predictive models is, as Dr. Jaffe said, the issue is not whether chemotherapy can hurt the cancer cell. It's rather a risk-benefit situation you have for the patient. How much total body harm do you have to give with your therapy to get the tumor to shrink and the patient to feel better? Because if the body is attacked by the therapy, that in the end is more toxic than it gives benefit. So we've been using organoids as ways to find the most efficacious therapy that don't hurt normal cells. We actually did a clinical trial that Dr. Jaffe was on the co-lead on, an international trial using such strategies to investigate chemotherapy. That is called the PASS-01 clinical trial that Dr. Jaffe and Dr. Knox is at University of Toronto. And as you know, Dr. Jaffe is at Johns Hopkins. They ran this international trial during the pandemic to ask which chemotherapy regimen, meaning gemcitabine Abraxane compared to FOLFIRINOX, might be better in patients with newly diagnosed advanced pancreas cancer. And the tissue came to our laboratory from all of the sites for us to ask, would the organoids predict the outcome of the patients? on the chemotherapy. And the clinical trial is finished and the first paper has been published by Dr. Knox and Jaffe, and is actually quite interesting to compare the two in a randomized study, which hadn't been done before in North America. And the organoids were very interesting too. It looks like the organoids are very predictive of several of the types of chemotherapy and how the patients eventually did. So that's important because it suggests that possibly these little models that you see on your screen might be a window into a patient's tumor. And we need to know that because, as mentioned, the RAS drug, for example, which, as Dr. Jaffe said, 90+% disease control rate, more than 30% disease shrinkage. Well, that's amazing. But it doesn't work forever, as she alluded to already. What might be something you could add to that RAS drug for that patient to make the effect so much bigger? And so that's the type of question that we're asking so that we can, as Dr. Rakeman said, make sure that we have the right people going to the right place, assuming the puck is going to be hit to us. So that, of course, we could give the puck to Dr. Jaffe so she can put it in the net. And again, sorry for the metaphors, but that's how we think of it. How can we move faster for our patients? They deserve more shots on goal. They deserve a better team on the ice. And Dr. Jaffe and I, we're managers and we have a lot of players. And Dr. Rakeman is making sure we have a lot of pucks. And you all are helping to make sure that the ice is hard, that we can skate on it. So we're quite excited because we have heard from the company that has this RAS drug that you heard about will be interested in supporting, in principle, the next version of PASS, which we would call PASS-02, since we lack in creativity. PASS actually is cool. It stands for Pancreatic Adenocarcinoma Stratification Signature. We're looking for signatures so we can game the market of the patient. And PASS-02 will be about how can we make RAS drugs better in a population of patients that maybe Dr. Jaffe can talk about. And again, I have to say these are preliminary conversations because I myself have to join one, I hope, this week or next week. But we're really excited because it gets us to this point where we might be able to help patients moving ahead much more quickly. So Liz, if you're comfortable, you can describe how that would work.
Right. So we're really excited. We just heard last week that Revolution Medicines will work with us. They'll partner. And so PASS 2.0, as Dave said, is going to be critical. So I wanted to point out one thing about PASS 1.0 that was the reason we did it. Patients, when they come to us with metastatic pancreatic cancer, we don't know which of the 2 or 3 now proven drug combinations are going to work best. And some patients do better with one versus the other. And as you can imagine, most patients, when they have metastatic cancer, they don't feel great. So we have really one chance to be able to have an effect for them and have them do better so that we can also enroll them into clinical trials after that. So the PASS study was really designed, as Dave said, to try to identify in different patients' tumors what might give us a lead as to which of the chemotherapies they should get. And it has been very helpful. So the next study we're going to do with the same type of format where we enroll patients throughout the United States and in Canada is to test the KRAS drug from Revolution Medicine. But we're going to move it to patients with what we call locally advanced pancreatic cancer. So the tumor is in the pancreas. The surgeons say they can't get it out because it's touching upon blood vessels or neurons. But if we could shrink it, they can go to surgery because when we look at their liver, we look at their lungs, we don't see any evidence that the cancer has spread to those organs. And so this is critical because right now we don't have anything for them. The chemotherapies don't usually shrink their tumors enough. But we are hopeful that by going to this population of patients, we can shrink it with the KRAS drugs. We have a window of 8 months to do that before those drugs stop being effective. And then we could take patients to surgery. So that's what we're going to do. But in addition, we get to, again, get their pre-treatment biopsy. And then if they go to surgery, we get the entire tumor and see how the treatment has worked. If they don't, we will get another biopsy so that we can see why the treatment hasn't worked. The last time we did about 130 patients, so it's a pretty large international study. We're expecting to do something similar for this study. As Dave said, we're still finalizing the design, but we are planning likely to compare the drug alone versus the drug plus chemotherapy to see if combining it with chemotherapy may be even more effective. Again, we don't know which will work better. We prefer just the drug since it's less toxic, but again, we want to do the best for patients. And so we have to figure out which way is the best way to give it in that situation.
No, I'm really glad that you both brought up PASS because I think PASS has been one of the most exciting things to come out in 2025. Not only because of the really exciting clinical results, not only because of all of the wonderful technologies that have been applied and organoids being developed, but also because the way it's changing what we think is possible in clinical trials for pancreas cancer patients. So the next question I'm going to ask you, Dr. Jaffe, relates to a question that came in on the chat, which is relates to something everybody on this call knows, is that patients can't wait. The current processes that we have are too slow for clinical trials, for drug approval, for things moving forward. So maybe, I guess the question, Dr. Jaffe, is could you, describe a little bit about what's different about the way PASS was designed from the setup of the trial that allows things to both move more quickly and to be more informative, and how that's, kind of feeding into discussions with down the line with regulators, with payers, with all the other aspects of the healthcare system to try to make sure that we are doing trials that address the needs of patients and approving drugs based on what's important to patients in the end.
Right. So, Andy, thank you for asking that question. So, one of the major concepts behind doing these multi-center trials is being able to get the different centers to agree ahead of time to contracting, okay, to sharing patient information, to sharing patient specimens, because each institution isn't experienced in every single scientific technology. And so we thought by bringing together sites, different sites that could access different patient populations. So people in Baltimore are different than people in Canada, Toronto, right? Toronto has more Asian Canadians. We have more African Americans. People in New York are different. People in the West Coast are different. So we're trying to make sure that we are testing our therapies in a whole range of culturally different individuals so we can understand, because their genetics are different, their environmental changes are different, and we know that they react differently to therapies. So again, doing this at multiple centers, getting all of the institutions' administrators to agree that we can share information. Again, remember, there's HIPAA, so that's always been a restriction. So we have to have good guidelines that prevent us from divulging information that would be detrimental to patients. We only want to share information that's going to be helpful in understanding how these treatments are working in different patients. And then also being able to have a system where we can share the biopsies. So we were getting 6 pieces of each individual patient's biopsies. And then we actually had a system where the first one would go to Dave's lab, the second one would go to Toronto. Because of different technologies, one piece would stay at Hopkins or come to Hopkins, depending on where the biopsy was done. Because we all had different ways in which we can assess the different, utilize the different technologies to assess the genetics, to assess the inflammation or how the immune system sees the cancer, to assess how drugs respond in the organoids Dave described. And we would meet on a weekly or every other week basis as a team virtually and discuss every single patient and all their data.
Mhm.
That was such an important meeting that we were having. We call that a tumor board, but basically we learned from every individual patient. Everyone's an individual. You're all part of the team. And I think this was critical, and that's why this was such a success. Now, this platform is in place. We can add additional sites easily because we have all of the administrative components that are able to work well among the different institutions. So I think now we can just continue to test new therapies on this, what we call platform a platform clinical trial. So we're very excited. We think this is going to even accelerate even faster what we learn. But again, multiple centers is critical because we enroll many different patients more rapidly and patients of different backgrounds.
Yeah, no, thanks, Dr. Jaffe. And I think it is that approach of having a platform that allows you to share the data, make decisions quickly, but also adapt and add in those next rounds very quickly and learn from that. And I think that's really the hope. If people got this, what's going to change from pass one to pass two is that adding in with new drugs available, with new approaches, taking those learnings, we can move very quickly through and start to test. And it's a little bit of a segue to the next topic I want to talk about is the approach that you've taken, Dr. Jaffe, within your own group very closely in this belief that the immune system is going to be a key component of how we control this disease ultimately. And through your steadfast belief that that's going to be part of the solution, and the way we're going to make that part of the solution is by really understanding the response of the immune system to the tumor in the patients. And so you've had this platform approach within your own group to try to drive forward vaccines and immune approaches there, where you've been seeing success by having that dedication to letting the biology guide. So maybe if you could, give a little bit of an update to the folks about, where we're going. You started a little bit in your introduction with vaccines and with immunotherapies, and where you see the potential of those entering into kind of the standard for how we think about treating pancreas cancer.
Yes, thank you for that question, Andy. So, again, just like we're learning a lot about pancreatic cancer and its biology, we're also learning about how the immune system sees pancreatic cancer. And this is critical because every cancer develops differently from the point of view of the immune system. And when I say the immune system, we're talking about inflammation. We now know that inflammation, particularly chronic inflammation, is definitely a risk factor for different cancers, including pancreatic cancer. It's a risk factor for lots of diseases, but specifically for cancer. But inflammation will be different depending on the organ in which the cancer develops. And also, the inflammation responds differently to different immunotherapies depending on the organ, but also the patient, right? So, the patient's genetics, the patient's other risk factors. So we've been using another type of platform study where we started testing an older version of our vaccine. It was a whole tumor cell expressing a protein called granulocyte macrophage colony stimulating factor, which we had identified more than 25 years ago is an important activator of the immune response that fights a cancer. It fights a virus, it fights a cancer. We tested this in patients who were going to surgery, but 2 weeks before, and then we get their tumor. So we get to see how the tumor has changed. Well, the vaccine definitely caused something called lymph nodes developing in the pancreas tumor, but they weren't activated enough. But we learned what it was that those lymphocytes or the immune cells needed to become more activated in these lymph node type structures. And so the first drug we added to our vaccine was anti-PD-1 because PD-1 is an immunotherapy now approved for other types of cancer where it helps the activated immune cell, the T cell, now actually function in the tumor. But it still didn't work very well. But we were learning that the T cell was asking for another signal to be altered, and that was something called CD137. And so now we've had a trial where we've given vaccine plus anti-PD-1 plus anti-CD137. And we've had now about a third of our patients having near total responses at the tumor before they go to surgery, which is critical. And they're doing much better post-surgery. Now, is that the answer to everything? No. We're now testing a new vaccine that targets mutated KRAS. And we've seen really good responses in patients who undergo surgery and then get the vaccine, and those patients are doing better long term. We're testing it in collaboration with the KRAS inhibitors because the KRAS inhibitors actually are able to modify a pancreatic tumor to allow the immune system to act better, to be more effective. And so we're trying to test that in patients. We've seen this happen in preclinical studies, but now we're testing it in patients. So hopefully next year I'll be able to tell you how that's working. We just started those studies. Again, these are Lustgarten funded studies. And then the final study that I think is critical, and I've seen a couple of questions already in the chat. Yes, these vaccines, when we talk about vaccines currently, we're talking about therapy. However, my biggest goal is to prevent pancreatic cancer. And so we currently completed a 20 patient study looking at safety of our KRAS targeted vaccine in people who had genetic predisposition to pancreatic cancer and small lesions on their pancreas. And we're seeing responses, immune responses against KRAS that have lasted now up to 3 years. That's as long as some of the patients are out. Totally safe, but in a smaller population of the patients who had lesions on their pancreas, we're seeing shrinkage. Again, we don't know what this means yet, but it's telling us that there may be some activity. We're now in a study where we're doing it in IPMNs, these big cysts that we can see, we can
Mhm.
biopsy. When they look like they're transforming to become a cancer, we take them to surgery. Well, what we're doing is we're getting a biopsy, we're treating them with the vaccine before they go to surgery because we do have some time, and then we get their cysts at surgery, and we can now start to analyze what the immune system is able to do or not able to do. We may have to improve on the vaccine. But again, long term, our hope is that we'll be able to have an optimized immunization approach, a vaccine approach that can prevent pancreatic cancer. And again, these are all studies that thankfully the Lustgarten Foundation has partnered with us on.
And it's super exciting, Dr. Jaffe. And a couple of things I pulled out from that to highlight for folks. It's again that same process of curiosity about the biology, really curiosity about what is happening with the immune system, understanding it within the patient, and not being afraid to fail and take chances and try new things that's led to success going forward. And success, you said it in quickly when you're treating these patients before surgery and you talk about near complete responses. To put a fine point on it, that means when the surgeon goes in, they have trouble finding the tumor. The tumor is gone or nearly gone, right? This is incredible. This is something that's unheard of. And now you want to not just treat the people that have small tumors, but stop those tumors from forming to begin with, which Dr. Susan is going to bring us to the next topic that people always ask us about. Dr. Jaffe is creating a challenge for us, right? She's going to be ready to treat folks with very small tumors, stop tumors from forming. How are we doing for identifying those patients with very early disease and getting towards early detection? What Linda in her introduction talked about a new partnership we have with Mark Foundation and AACR and others to look at this, but that is a challenge ahead for us to think about early detection, correct?
Yeah. So all the excitement you heard from Liz about the advances in immunotherapy have, as Andy was saying, are based on over 3 decades of Liz tirelessly pursuing this target and this topic in the face of adversity and lack of funding from the government, et cetera. The early detection of pancreas cancer is similarly a very challenging topic. It's one that is self-evident and that many people have spoken about over the years. We don't have a test today to identify those patients unambiguously besides high-resolution scans that would see a tiny tumor, usually radiation-based scans, such as a CAT scan. There is the magnet where you look at water content and its properties, which is the MRI. But again, what we're trying to find is a tumor that's smaller than your pinky nail. So imagine less than a centimeter in size. Those tumors are usually found accidentally. Patients are getting a scan for another reason. They go to the emergency room because, again, they have some other symptom. But if you can find a patient that has a very tiny primary tumor, oftentimes you can help them to a great extent. They will be a candidate for surgery and medicines either before or after the removal. Combining immunotherapies for such patients would obviously be very exciting. And I know Dr. Jaffe can't wait to have her clinic full of patients with stage 1 pancreas cancer, or better yet, pre-stage one pancreas cancer, the people who would otherwise develop this tiny tumor. So how might we identify these patients? Now, you've all heard of the blood tests that have been developed and we use to monitor patients that have pancreas cancer. Can we apply such tests to people before they have cancer? Well, this has been a question that the field has worked on pretty actively for, again, more than 3 decades now, probably 4 decades. So far, we don't have a simple blood test, for pancreas cancer in this fashion. We do have clinics where patients at high risk for pancreas cancer are followed very carefully. That includes blood tests and imaging tests, including an endoscopic ultrasound, where again, we're able to pick up some patients by this modality. But because we knew this was a hard problem, as Linda mentioned in the beginning, we partnered with other organizations to have a think tank on this issue and bring experts in who think about pancreas cancer and other types of cancer, including engineers, mathematicians, physicists, and medical scientists and medical doctors. We've actually had some very exciting proposals come to us through such a partnership, including several from people who want to study the health of the pancreas. You may say, why bother studying the health? Well, what we have found by talking to lots of folks is we don't know as much about the health of the pancreas as we thought. The pancreas' job is to help you digest food and balance blood sugar. And when those things don't work, people have symptoms. If they can't balance sugar, they have a version of diabetes. If they can't digest their food, they have malabsorption and they lose weight because of that. Well, we were wondering, could we actually measure the health in people who don't have cancer and actually come up with better tests that would tell us on the spectrum of extreme health to extreme disease, who should we be worried about that would be developing pancreas cancer? The reason why is that when someone has a pancreas tumor, the rest of their organ doesn't work very well. It poisons the rest of the pancreas. And so we actually have a project now we're funding because of our partnership with the Mark Foundation that's allowing us to go after this topic. We're hopeful that such projects will identify patients early before they have pancreas cancer and that we can have them go to trials that Dr. Jaffe can organize to see if we could maybe prevent them from having pancreas cancer. We'll have to see. But it's quite exciting. And it's one of, I'd say, the main challenges we have right now. Can we identify people early so that we could either prevent or, quote, intercept the disease and use medicines like immune therapies because that goes after not just one type of pancreas cancer, but many at the same time. And that's a way to overcome what we call drug resistance in the RAS space. The immune system has multiple T cells that goes after the same cancer cell. It actually has it all built in from the beginning. So that's, my perspective on early detection, massive problem. We need support from the community so we can get there because we're not there yet. But we have some extremely talented people that the Lustgarten Foundation has now informed and touched that, we are looking forward to supporting, pushing this forward.
No, I agree. This is definitely one of the biggest challenges we're facing right now, something that at times can feel impossible, but we've also spent this afternoon or this midday talking about challenges that used to be thought of as impossible. Could we develop drugs that target KRAS? Can immune therapies work in pancreas cancer that we're chipping away at now and seeing hope for? So I think if we go back to our cycle of curiosity to patient need to bringing in new approaches and innovation, we're sort of at that point on early detection of now's the time, as you pointed out, Dr. Tuvison, to bring in the engineers and the mathematicians and the people who think about a problem differently to help us to bring in some innovation and make progress here. And hopefully this won't be a decades-long challenge, but we can shrink that by ramping up the effort that we have here. So I'm watching the time here. I think we could go on and continue to talk for hours and hours about the exciting things that are happening. There are some questions coming in that I think we're trying to speak to most of these as we go. including I think one of the things you just alluded to, Dr. Tuvison, working with other cancer types. And there are areas like early detection where we can learn from other hard to detect very deadly cancers like ovarian cancer and glioblastoma, where we share some of the same challenges. Um, but maybe before we get Linda back and start to wrap up, I'd like to give each of you a chance to kind of close us out and talk about, obviously we talked about some great progress. We're hopeful about things moving forward, but there's lots more to do. So kind of what's exciting you today for the future, and what do you hope a year from now when we meet again, we'll be talking about will be the great advances that we're driving forward with. So maybe we'll start with you, Dr. Tuvison.
Well, our job a year from now is to have PASO 2 open and accruing patients. Dr. Jaffe and her colleagues are excited to get that off the ground because we need to identify additional agents that we can add to these RAS therapies to give the most profound, sustained, and deep response in patients. Our job is to put ourselves out of work, right? So that's the goal, and have all you and your loved ones be healthy. We have to come up with better therapies. Being able to combine it, obviously, with the skills that Dr. Jaffe has in the immune therapies is going to be a big part of it. On the science side, understanding why we're having this trouble with the RAS drugs as a sustained medicine will be important. And following this thing called heterogeneity in the tumor, where you have a wolf with many coats in someone's body. And that's really the science of what we're trying to understand. And then a year from now, in the early detection space, hopefully we've identified some people that are bold enough and smart enough that they can start to tackle this problem. And we can report back to you on how that's looking. So that's my hope for the coming year. And I hope it's commensurate with Dr. Jaffe's.
Absolutely. I think I'll just add where you didn't add yet, and that is that I think the first KRAS inhibitor will be approved by the end of 2026. I think that others will follow pretty quickly, so we may actually see another one by the time we talk next year, or another one that's close. I think we're going to see many studies that have combinations of these KRAS inhibitors that will be enrolling patients to try to overcome the resistance that Dr. Tuvison has talked about. And I hope I can give you more exciting news on how immunotherapy is doing better for patients with pancreatic cancer. I think we're at a good time. We're not moving fast enough. I get that. I really do. And I feel terrible. We can't move faster, but I have to say we're moving as fast as we can. And I think we're moving faster than we've ever moved before. But of course, that doesn't help those patients who need it today. But anything we could do to help, we are trying. We really are. So thank you all for joining us.
No, and thank you, Dr. Tewes and Dr. Jaffe. And the one thing I will add on top of that is we didn't get to where we are right now because, some smart people sat alone in a lab and toiled away in darkness, or because one person had a brilliant idea that they, brought forward as a lone entrepreneur. It's really because a community drove this forward, whether it was, patients and families standing up and saying enough is enough, we need to do more, that led to the start of the Lustgarten Foundation. Because people come in and bring their resources and their time, and patients participate in trials that helps to drive things forward faster. And even as you just pointed out, Dr. Jaffe, we were hopeful and we expect to see the first approval of a rash drug probably ahead of schedule if it happens, because people stood up to the FDA and said we need to move this faster. That allowed the FDA to come in and put a fast track and a voucher to up the priority of the review of this drug. We'll still have the same quality of data and safety data, and will be evaluated with the same amount of rigor, but will be given a priority because everyone recognizes that pancreas cancer patients in particular can't wait, and that we need to see this going forward. So I know I'm excited. I'm excited both because of the progress and because of the work that we have to do, and that we really need to keep pushing forward. Linda, I hope you share in the excitement here, and that the wonderful job that Dr. Tewes and Dr. Jaffe do, both in helping to guide us and to help us to really understand where the field is going.
For sure, Andy. Thank you. And thank you, Dr. Tuvison, Dr. Jaffe. Your excitement is palpable, as is your persistence. And we are so grateful for your patience. We know this has been a long haul, and but we can see it. It's right there on the horizon. And so our title today was Turning Hope into Expectations. And as our community heard today, we expect that by the end of the year, this year, we will have a lot more good news to share with our community. Throughout this past year, Lustgarten has stood up for science, and we will continue to do so in 2026. At a time of funding threats and uncertainty, the foundation became a national voice advocating publicly in supporting early career scientists, investing in diversity and equity, and ensuring that progress benefits all patients. Because of our researchers, donors, advocates, and partners, hope is giving rise to expectations, and grounded in progress, and that science is advancing and our community is mobilized. During November and December season of giving campaign, we received more than 15,000 gifts from 13,000 donors, totaling $7.5 million. So a heartfelt thank you to all of those of you who joined us today and to our Lustgarten community in the United States and around the world. We thank you. And finally, I'm looking forward to our third International Women's Day Symposium at Oregon Health and Science University. This important discussion takes place on March 10th and is co-chaired by Dr. Jaffe. Thank you, Liz. And renowned scientist Dr. Lisa Cousins will be her co-chair. And together, we're bringing some of the most brilliant female minds in pancreatic cancer research to Oregon. Looking forward to a really a productive conversation in March, on March 10th. Registrations opens February 4th, so check your inboxes. Thanks to everybody. We really appreciate all you do for the foundation and for our community and for patients. Thanks, everyone, and we'll see you on March 10th. Thanks, everyone.